Helena Kandarova. Helena Kandarova, she's a distinguished scientist in the field of toxicology. She's particularly known for her work in vitro toxicology and alternative to animal testing. She serves as the director of the Institute of Experimental Pharmacology and Toxicology at the Center of Experimental Medicine of the Slovak Academy of Science. She also established and led the MATTEC in vitro science laboratory in the European Union. And her research is mostly dedicated to understanding the toxicological effect of chemical drugs and medical devices on epithelial tissues, with a strong focus on regulatory toxicology, and we see that today. She's had about 20 years of experience, and she significantly contributed to development, validation, and implementation of animal-free testing method. And she always did a great effort to work on the inclusion of these alternative methods into regulatory frameworks, such as European Union OECD and guidelines for ISO standards. And she also actually is the president of the European Society of Toxicology in Vitro, which is ESTIV, also vice president of CTOX, and chair of the Slovak National Platform for 3R, among other responsibilities. She has also been recognized as a national expert in OECD expert groups and representative in different international toxicology networks. And she also received a number of prizes and honors, including the Bjorn Ekwall Memorial Award and the Dorkamp Zimden Award for achievement for promoting alternative to animal testing. So, Helena, the floor is yours.
Thank you very much, Mark, for this kind introduction, and thank you everyone for joining today for this webinar. I hope that you can hear me well and you can see me well. This presentation is available afterwards for sharing, so if anyone would need it, it will be available through the EUROTOX Secretariat. So my presentation today will be about the issues with reconstructed tissues, and I will be guiding you through some interesting aspects related to the work with the 3D tissue models. As Mark already said, I have quite a lot of experience from the area of the regulatory toxicology. So just very briefly to show you my involvement in all these validation trials from a very young PhD student to a manager. I have been involved in the skin irritation and skin corrosion validation studies that led to the OECD test guideline 431, 439. We worked almost 20 years for the development of the phototoxicity testing guideline that has been adopted a few years ago. We were also working on the development of the eye irritation protocols, protocols for the medical devices testing, and many other things that are not today's scope of the presentation, but they are also contributing to the experience with the 3D tissue models and the validation and regulatory aspects. So let's speak first about the technology and the 3D tissue model itself. Some people think it's relatively new. With these 3D tissues, we hear that it is an innovative science and so on. I would say that specifically here, the reconstructed human skin models is not a new technology, and it has been available since 70s of the last century. The initial idea to have 3D reconstructed human tissue model came from the need to treat burn patients. And the first reconstructed human tissue model started to be developed around early 80s. And in the 90s, there was an idea that these technologies could be most probably also reducing or replacing animal testing. Some of the very first models that were made available around 1993, 1996, the test skin and the skin square models provided a lot of interesting data sets, but unfortunately they were discontinued. And it was not really encouraging for the people that were working with these technologies and developing protocols to have a technology and that would disappear in a few years. But fortunately enough, their successors were more successful in commercializing of these 3D tissue models, and the very first technologies that appeared along with the older models remaining on the market. And these were the EpiSkin model, the Skinetic model, and the EpiDerm model. The first tissues were relatively simple, but later on, around 2000, more complex tissues started to be manufactured. And there were many other tissue models that have been commercialized by other suppliers, other companies worldwide. You can see examples of this technology here. It is not a full list. It's just to show you that basically we have two basic types of the reconstructed skin models. The first one are the epidermal tissue models. They are composed mostly of the keratinocytes that are seated on some membranes. And they are more or less similar. They may have a different thickness of the stratum corneum, but with regard to the barrier properties, if they are developed well, they are quite similar. We have also full thickness models, and the full thickness means that it has another layer that is more complex, usually contains collagen and fibroblasts. These are the pictures here below. And with regard to the complexity of the tissues and level of the technology readiness, they may provide significant differences in the barrier properties and the organization of the various layers. There are on the market many more of these tissue models, and it is important really to check what they are able to provide. There is also a very important new area emerging, and these are disease models. Disease models are something that is not so easy to develop because the diseases are quite, I would say, complex, and every patient is slightly different, especially in the diseases like psoriasis, like atopic dermatitis, and so on. But there are models that are addressing also these needs, although I have to say that there are more hospitals that are interested in development of these tissue models. However, I would like to say here, there is a very nice consortium called NetSkin Models, and they are really focusing on this type of the models in their project. And we hope that out of this collaboration, we will see in the future more tissue models that can address also this emerging area. With regard to the technology of the tissue reconstructions, basically there are two basic attempts how to prepare these tissue models. You can grow the tissue models on cell culture inserts, or you can grow them on the biological scaffolds. Most of the companies that are selling the tissue commercially are working in the cell culture inserts, but there are exceptions. And those tissue models that are grown on the scaffolds are usually models that are used for some advanced studies, like the efficacy of the drugs, skin aging, and so on. So for the simple toxicology assessments, the cell cultures on the 3D tissue membranes are usually sufficient. So let's speak about how these tissue models are basically constructed on the 3D tissue model. So usually you have the insert with some kind of the membrane that is semi-permeable. It can be Teflon, polycarbonate, whatever. And these membranes are covered by a material that enables growth of the cells that are collected from the donors. There are three basic types of the cells that we collect: fibroblasts, keratinocytes, and also melanocytes can be very easily included. So the cells are seated on the membrane. It requires to have a good composition of cell culture media that contains some growth factors, and also you need to meet some specific needs for the growth factors that are needed for a specific type of the tissue model. So for the skin, you usually have there the keratinocyte growth factor. You may add hydrocortisone. It can be vitamin C added into the media. It can be a mix of different lipids that are added into the cell culture media, and so on. But this is not all. If the cells are doing well in the media, at a certain time, you need to remove the cell culture media and expose the cells to the air. And this type or this step is called cultivation at the air-liquid interface. In this step, basically the cell layers are reacting on the changed conditions. The skin model is stressed, and basically the cells start to create the stratum corneum. That is the barrier, basically, of the tissue model in combination with the lipids in the stratum corneum. We also do have more advanced ways how to manufacture these 3D tissue models, and this is the 3D bioprinting. It's an innovative technology that is combining the use of the cells with the biopolymers, also called bioinks. And these bioinks are enabling us to accelerate the production of these reconstructed tissue models, especially in a case we are creating more complex structures that, for instance, contain the dermis part. If you culture skin models in a classic way in the cell culture inserts and you do it manually, it requires for the full thickness model 6 weeks on average. If you bioprint the matrix, it can be considerably shorter time. The bioprinted matrices are usually more stable. They don't undergo the shrinking process that is caused by the presence of the fibroblasts. And overall, if the technology is well developed and the composition of the culture media is okay, then the skin models are also working or having good properties. So you can see on this picture the bioinks with the different cells that are basically used to create this model. On the other hand, if the matrix is prepared, the skin model is seeded with the keratinocytes, and it still requires time for the development of the typical cell layers of the epidermis. This cannot be bioprinted. Sorry, there is some automation in my presentation. It's moving the slides forward. So the bioprinted tissue cannot be bioprinted in one day with all the features. The skin model really needs some time to develop. So once you prepared your matrix and you edit keratinocytes on the tissue model, you still need some time, something between 7 to 14 days to fully develop the tissue model. As I said, the advantages of this tissue production is that there is a minimal lateral contraction observed in the bioprinted samples, and the samples usually have higher tear values. Tear is a parameter that is telling us something about the tissue barrier, and the tear is usually higher than in the samples produced in the classic way. If you are interested about the bioprinting of the skin, there is a fantastic publication from 2018 and 2019, and I really encourage you to have a look into these 2 papers, from which also the pictures were taken. Of course, we do have also a very innovative we are trying to mimic. The most easiest way or most easiest barrier that we are mimicking with these 3D tissue models is the physical barrier. This is basically barrier model composed of the keratinocytes or corneocytes and the lipid of the cornified envelope. It's kind of brick and mortar structure. This can be mimicked very easily. We are able to grow cells very nicely into these structures. The chemical barrier is also something that is present in the reconstructed 3D cultures. The chemical barrier contains the antimicrobial peptides. We do have the radical oxidative species scavengers in the human skin. That all works well. The problem starts a bit when we try to mimic the immunological barrier, because we are usually lacking the cells responsible for the immune response. And many people that try to combine lung-enhanced cells or T cells with the 3D tissue models had problems how to keep the two other barriers functional. But there are these days also tissue models containing that type of the cells. So there was a progress done compared to the, let's say, last 10 years. And a real issue comes when we want to mimic also the microbiological barrier and the microbiome. Because there, a big problem starts that the microbiota, when it overgrows, it kills the skin. And usually, as you know, if you work with these tissue models, they are coming into your laboratory sterile. And if something is starting growing in the media or on the tissue, you usually have a problem. If you want to introduce the microbiota into the 3D tissue models, you need to approach the skin model producers and tell them to remove antibiotics from the media and from the tissue model. And the production, of course, is then more expensive and also more challenging. So the type of the microbiome research on the skin, I don't say it's impossible, but it's challenging and a bit more complex than if you just simply look into some toxicity or some claim confirmation about the efficacy. There are many procedures and many tests which you can utilize to check the barrier properties, and I will not go into the details, but the most important things to check is the histology. Histology already tells you quite a lot about how the tissue may react into some chemicals and also tells you a bit of the barrier properties. We are usually also combining the histology with the functional check, so-called ET50 assay that tells us about the barrier robustness. You can, of course, depending on your research, screen for the knockout genes. You can evaluate the tight junction functionality, cornified envelope composition, and many other things that you would be able to do also on the native skin. Yeah, there are many attempts how to improve and modulate the barrier properties by the skin model producers. And of course, if you are developing your own tissues in your own laboratory, that you know that growth conditions, as for instance, temperature, days in culture, and oxygen levels, and even humidity, may very much influence how your 3D tissue models looks and how it will develop. The growth factors and nutrition in the culture, this is also something extremely important, because if you modulate the calcium levels, you add or remove vitamin C, hydrocortisone, and so on, you can achieve completely different structures in the 3D tissues. One thing that is also quite well known, especially for those that are using 3D tissue models supplied from suppliers, commercial suppliers, the type of the semi-permeable membrane matters. There are usually two types used, which is the Teflon or polycarbonate. And very crucial is the size and the distribution of the pores in the membrane. Some of the membranes also may have different chemical resistance. So if you are working on your own tissue models, please check all these parameters that may influence the quality of the tissues you are developing and you are working with. Yeah. Then the barrier properties also can be influenced by some other things that are not as easy to control, which is the cell donor age, exposure of the donors to the xenobiotics. That means your donor might have been exposed to some drugs and environmental factors that you don't know. What you can influence is also the skin origin from the donor. You can choose. It can be either foreskin or abdomen. And the cell origin in the skin is also something to consider, because there is a proven record of the fibroblast growth. There are differences if you take fibroblasts from the papillary and reticular regions of the dermis. And of course, the passage number of the cells used to construct the reconstructed human epidermis really matters. You know that most of the tissue models are prepared from the primary keratinocytes, and you cannot really subculture them more than maybe 3 times. So if you are playing with the barrier with the same donor and with the same more or less insert, you can achieve very big differences just by, changing one or two components in your tissue model. This is coming from my PhD work, and it is a tissue that came from the same supplier with the same donor, but there was some difference in the days in the culture before shipping, and also the composition of the cell culture media. And it was immediately visible on the ET50 values used as the parameter for the quality that we have big differences between the tissue. So the first tissue was able to resist to our cytotoxic marker 6 to 8 hours, whereas the other one, that was a tissue that was much more resistant, and we had to wait for the ET50 value 10 to 12 hours. So this is also something to consider when you are working with the tissues. Please work with the tissue models, ideally, let's say 24 hours after the arrival of the tissue models. Don't store them in the incubator until next week, because you may significantly modify the barrier properties of the tissues. Yeah, many questions are coming to me, like, is it better to have commercial or homemade tissue models? So I would say if you have commercial tissue models, you can rely basically on the quality certificates. So every batch of the tissues are coming with something what we call birth certificate. It tells you what was the barrier properties, what was the optical density of the tissue in the test, and so on. And these tissue model suppliers, they also have very good record on these parameters. You can see it here on the picture. And you know that you are getting more or less standardized product every month, every year, every 10 years. With your homemade tissues, of course, you know what is inside. You can play with multiple donors. You have multiple choices of the inserts, of the things that you add to the cell culture media, and you have also more flexibility in planning and conducting your own studies. On the other hand, you have a really high risk of non-donor related variability due to the less stringent requirements for the biological material, cell culture media, and production components. And also, if you are working with students, you know that they are very hard to get to adhere to some good manufacturing practice rules. Yeah, sometimes it can lead to real disaster in the laboratory. And well, how much of similarity or differences we can see? I can tell you with the commercial tissue models that are in the OECD test guidelines, the variability is really minimal. There are some differences, let's say, in the surface or in the thickness of the stratum corneum, but the barrier properties are really very similar. On the other hand, it's your task to evaluate your supplier. So ask for the data, collect the data, do your own testing to check that the batch that is in your hand is really matching the well-established standards that the skin model supplier claims. So let's have a look on the tissues used in the in vitro toxicology, and I will be focusing quite a lot on the regulatory toxicology applications in two different fields. First of all, this is a short overview on where the skin models were implemented at the levels of the OECD and some other regulations. So we have validated regulatory accepted models for skin corrosion for a really long time. The first validation studies and implementation was happening in 2000 for the skin corrosion. The model was implemented into the European regulation in 2000 already, and at the OECD it was in 2004. It took quite long until everyone was happy with the guideline as a product, but you see, 20 years ago, there was a method already in the guidelines. Later on, it was the skin irritation test with multiple skin models. It was endorsed by the OECD. It was also implemented in the transport regulations, and we also recently made a modification for the use of the skin irritation protocol and 3D tissue models within the medical devices area. And there's a new ISO 1099323 that I will mention a bit later. We do have also phototoxicity test. It is validated with the epiderm model, and this one, it took 20 years from the initial development until the implementation. And really the method was there already 20 years ago, more or less in the finalized form, but it took really long until it got implemented into the regulatory frameworks. For the skin sensitization, there are not yet regulatory implemented tests, but there are projects running at the OECD. So we do hope that in a period of 2-3 years, we will see also test guidelines in the same skin sensitization area. And the same is valid also for the genotoxicity areas. For the skin penetration and absorption, the guidelines that are already in place, they don't really mention the 3D tissue models, like reconstructed 3D tissue models, but they are used, for instance, for studies of baby skin products, because this reconstructed skin is very sensitive. So if the product is safe on that type of the skin, it is assumed that it will be safe with regard to the penetration and absorption also in the kids. So we don't have information yet whether 3D tissue models get adopted into this type of the guidelines. Validation is, as you know, quite a complex process. And here you can see the classical way how we validated these tissue models in the past. It may take really up to 20 years until method gets implemented. And it is the sad reality that unfortunately it takes so long. But also technically, sometimes, you know, methods development is not finished properly, and laboratories are stepping directly into the validation, and they omit pre-validation steps, and they are then disappointed that the model didn't work well, and they had to go back into the development of the method and repeat the validation. And the review process also sometimes takes very, very long. Just some discussions may take 2, 3, even longer years. And it takes some time until the regulatory acceptance is achieved. But there are attempts how to change the process, and we hope that in the next 10 years, we will see much more flexible validation procedures, especially now when we have a lot of these tissue models implemented in the regulations. When you are working with the 3D tissue models and developing specific protocols, you have to consider that the predictivity of your in vitro model is always linked to the in vitro method design and the real life exposure. So you may have the same 3D tissue models for various purposes. As you could see, we have the skin irritation, skin corrosion, phototoxicity with the same tissue models, but we need to fine-tune the exposure conditions and maybe sometimes those per area aspects to get the requested sensitivity and specificity, overall predictivity for some specific group of the materials. I will show you here on this example what I mean. In the OECD test guideline protocol 439, which was developed to test neat material, neat chemicals, we had a protocol that works with relatively short exposure time, takes 15 to 60 minutes. And then there is very long post-exposure period, 42 hours, to allow development of the cytotoxicity effect after we remove the material from the tissue surface. And the endpoint is MTT test, which is very standardized. In this protocol, we are able to get information about the hazard of the material and about its possibility to cause skin irritation of grade 2.3 and higher as compared with the animal. If we want to use the same tissue model for the testing of medical devices, we need to work with very, very different exposure conditions, as you will see later on the example. Because if we would use the OECD protocol, almost all extracts from the medical devices and some gels that are also classified as medical devices would be negative, and everything would be perfectly safe. So to achieve really increased sensitivity, we need to go to completely different exposure scenario, which takes from 18 to 24 hours, and it also has a higher dose of the material. And only afterwards we are checking the viability or interleukin-1 alpha release. So you see the same tissue model, completely different conditions, but still both of the protocols are highly predictive of the skin irritation of the materials that are tested. Now, I just would like to show you some example of a validation, typical validation from older times, which was the epiderm skin irritation validation study, and some follow-up activities that led at the end to the successful implementation to the OECD. There was in 2003 really a specific problem to solve because in pre-validation phases of this big validation exercise sponsored by ECVAM, all the skin models failed. And the skin models developers were asked to redevelop the protocols to increase the sensitivity and specificity. So in 2001 and 2, L'Oreal developed a protocol for the EpiSkin model. And in 2003, we were transferring this protocol at ZBET, it's a German organization, to the epiderm tissues. And there was an agreement that we will try to come up with a common protocol for both tissue models, which would also improve the possibility of the regulatory implementation of these protocols if successfully validated. So we basically didn't change much in the protocol between the epiderm and episkin and went through the first phase of the validation study. First phase was looking very well. There were really minor differences with the 20 materials that were tested. But in the second phase of the validation study, where we increased the set of the materials, I think it was 60 samples that we tested, suddenly one could see big differences between the two tissue models. And the EpiSkin assay provided data that were close to what we could see in the rabbits, whereas the epiderm model was highly specific, yes, it was highly specific, but the sensitivity was low. So it was missing some of the irritating materials if we compared this prediction to the rabbit data. We had human data in hand and they were quite favorable for the epiderm model, and we could explain by this human data the problematic cases, but the human data were not accepted at that time, and the decision of the management group was that we need to make the epiderm protocol more sensitive. So this idea of the common protocol was not 100% working in this type of the case. When we were investigating what was going on, we had an analysis of the animal looked into the human data. And as you can see on this picture that is showing you the data set for 45 substances tested in 3 to 6 animals, you see that there are some materials classified clearly as non-irritative, non-irritating. These are the blue dots. Then there is a group of the materials that is relatively clearly irritating group, the high scores groups with pink, 3 to 4. But there is a really gray zone here, you can see, where the predictions are basically in some animals same for irritating and non-irritating. The whole prediction model in animals is just based on some artificial calculations, and sometimes, you know, the material falls into the irritating and sometimes into non-irritating area. So it was clear that the animal test is also not so great to predict what we need to predict. And it is really hard to fine-tune in vitro protocol to this type of the data set. Moreover, I had mentioned that we had also human data in hand. So we compared, we had a comparison for specific materials on the rabbit and in the human volunteers, and we did also some prospective testing, and we were able to confirm that most of the materials that were from the epiderm validation falsely negative were not irritating to the human skin. But as I said, it was not accepted at the time that human data could be used as the evidence, and we had to redevelop the tissue model to be closer to the rabbit predictions. So we were not redeveloping the tissue model, but the protocol. So what we did was a significant prolongation of the dose per area and the exposure time from 50 minutes to 60, and dose per area was also increased. And basically with that small modifications, we were still able to follow the idea of the common protocol, but we really needed to be specific for the barrier that was in our hand. We learned quite a lot from this exercise. We learned that although the idea of the common protocol works for the 3D tissue models, the barrier properties of the model needs to be checked and considered carefully. We also learned that rabbits are really overpredictive models. And as I said, we conducted some follow-up studies where we confirmed data produced by David Basketter at Unilever, and it was also even published. And from these exercises, there were many laboratories that learned how to do the catch-up validation studies and save time and money when they were developing their own protocols for their own tissues. So it was a very good exercise. It took some more years for the EpiDerm to catch up with the OECD implementation, but we succeeded in 2009. Since we had a success in the chemical area, there were groups that started to be interested in the concept of skin irritation testing of medical devices. You know, medical devices are very, very broad group of things that you can basically implement or use in patients to improve their conditions. And it is also complicated to test the irritation, cytotoxicity, or sensitization of these materials because they are so complex. But they are the big three tests that needs to be done, and irritation is one of them. The complexity of the testing in the medical devices area is also linked to the way that you basically don't test the medical device itself on the skin, but you usually test extract from this medical device. And here the complexity starts with the test materials. You can do different types of the extractions. You need to extract into the polar and non-polar solvents. And the guidelines are telling you that you have some possibilities how to extract depending on which regulation you want to meet or which sterilization process the medical device undergoes and so on. And there are also some deviations also, whether you need to extract only 24 hours or 72 hours, whether the extract needs to be shaken or not. And all this is very much influencing the prediction of the toxicity at the end. So to make it short, when we were approaching the validation for the medical devices, we tried to harmonize the way how the extracts from medical devices were prepared. And we knew that in the medical devices area, the skin irritation test that it is preferred, especially in the United States, is a test for intracutaneous rabbit test. In that type of the experiment, you are injecting the extract from the medical devices below the epidermis, just in the area of between the epidermis and dermis. So it's intracutaneous test. And there, it really doesn't matter whether your skin has good or bad barrier properties. It's basically advanced cytotoxicity test. So in this type of the testing, if we would apply a protocol that was developed for the OECD chemicals, it would not work. So as I said at the beginning, we had to change the exposure conditions to be able really to soak the tissue model in the test material. That's why we have such a long exposure period times, 18 to 24 hours. And we also increase the dose of the test materials. Actually, this type of the protocol is very similar to approach that is suggested for testing of cosmetic formulations. That protocol is described by Claudine Fowler in her paper from 2002. The only difference is that for the cosmetics, they are using lower amounts, usually between 20 to 30 microliters, because there the skin barrier matters. You see that replacing of this test would save a lot of pain and suffering to the animals, because you see how bad the areas after injection of some cytotoxic materials may look here. This was an injection of the sodium lauryl sulfate. It was not done by us. It is a historical picture that was provided to me for this purpose of the presentation. So also here you see that we really try to replace a test that is causing a lot of suffering to the testing animals. The group that was working on this project, I will not mention all of them, but it was a large project that took from 2010 until from 2011 to 2018, and there were many laboratories participating in this. I'm very thankful to, especially to Kelly Coleman and Helmut de Jong for their guidance and for their insights into this type of the validation. And you can review all the work that came out from this project. It was also awarded at several meetings. And the result of this is that finally, we do have ISO guideline that replaced this type of the animal test. It was adopted, I think, in 2021. But it is still not yet accepted everywhere. So it is accepted in Europe. It has been implemented also in India and some other countries, but the US FDA is still hesitating to fully adopt this type of the procedure. So there is some work that still needs to be done, especially in the United States. And I am really happy to inform you that there are follow-up projects running in the area of skin sensitization, and I do hope that we will see more coming out in form of the publications and hopefully also the regulatory implementation of this test in the future. So let me conclude. I touched very briefly the area of the irritation, inflammation. I also mentioned photoirritation briefly, but there are many, many other areas where the 3D tissue models can be used, and this is the genotoxicity, as I mentioned, the disease tissue models, wound healing, microbial infections, and drug penetrations, for instance. So you see that this is the 3D tissue models are really a nice tool that can be used in the toxicology, and there are very nice opportunities with the developing technologies like 3D bioprinting, omics, and chip technologies. And I hope that these things will be implemented soon. I also say that we will need a new generation of scientists that are willing to work on the validation studies and implement that into the regulations. And if you want to see your test model and the test protocol in the regulations, you should start immediately after PhD or even during the PhD, because it really, really takes long. But at the end, when you see something like this implemented, and you know that it is a result of your like 10 to 15 years work, it is really very nice feeling. These tools basically not only enhance our testing possibilities, but they are really useful in replacing the animal testing. They are reducing the animal testing that is needed, especially in the area of the disease model, where the animal models are not really predictive. I do have big hopes that the disease tissue models will help us to address some problems that are just basically increasing. You know how many atopic dermatitis we have, psoriasis events, and it's constantly increasing. A good sign or good message that I can leave here is that we have these methods implemented into the OECD test guideline. They were also implemented, for instance, into ICH guideline on the phototoxicity. And we do see the recent implementation into the ISO standards. And I know that there are many exciting projects at the OECD that will bring new guidelines in the near future. The 3D tissue models are very useful also to understand the pathophysiology of the events that are happening in the skin, especially if we will be successful with the development of the disease tissue models. And as I said, the new technologies may bring much more advanced skin cultures than what is currently in our hands. And I do hope that all this will together open doors for the innovative animal-free drug development and testing, and also the use in the personalized medicine. And this was basically the last word from me. I thank you for your attention, and I do hope that we will see each other in some meetings. And I would like to use this opportunity to invite you to the ESTIF Congress that is happening in June, 3 to 6 in Prague. And if you are interested to join, we do have still the early bird registration opened, and by this Friday you can still deliver your abstracts. Thank you very much for your attention. And now I'm ready to answer the questions if there are some.
Perfect. Thank you very much, Helena, for this very complete and very detailed presentation. So we have some questions. So I will start with a question from Anna. So there is 4 questions. How different are these models from those used in cosmetics? That's the first question.
Yeah, so cosmetic industry is using these tissue models for, I would say, 25, 30 years already. As I said, one of the very first models were developed under the auspice of L'Oreal at the EpiSkin laboratories. Also, the technology of the skinetic model, I know, is coming from a PhD study at L'Oreal. So basically, these 3D tissue models were co-developed by many of the cosmetic industry labs, and I know that they are using them quite a lot for the safety assessment of their products, like companies like Unilever, Mary Kay, Beiersdorf. You can find multiple, in many publications where these things are used. So they are not really differing, but for some specific things, you need to consider more complex tissue models, like if you are interested in skin aging, for instance, you don't take the basic 3D epidermis model, but you take a model that is constructed for that specific purpose. For instance, the Finion model at Henkel, that is a model that is also suitable for some advanced studies with regard to the, I don't know, aging, antioxidation, and some other things that they need to test.
More questions. So are iPSC regulatory compliant, or only primary cells are used for the reconstruction?
Well, the models that are currently in the guidelines, to my best knowledge, are all primary cells-based models. But of course, if a model constructed from iPSC cells, basically, if they fulfill the requirements that are set there, and if they are validated for that purpose, I don't see any reason why they should not be compliant and useful. So yeah, it just needs to be validated.
Okay, another question, quite technical. What kind of matrix do you use for the cell growth?
Okay, there are different types of the matrices. Some of the 3D tissue model producers, they don't use any of the matrices because they say that the cells are able to produce what they need on the membrane. Some other companies are using collagen. Some other companies are using some alginates. There are also biological scaffolds that are more complex matrices, but I cannot really tell you all the details because these are manufacturing secrets.
So there's another question that I think already answered. Are the models used for other types of medicine other than chemicals one? I think you already touched base with the medical device.
Yes, yes, we can use that in the drug development and efficacy testing of the medicines, of the cosmetics, without any problems.
So now the question from Nathan Stockman. Thank you for the presentation. Why using reconstructed models instead of skin explants, like in the absorption guideline of the OECD 428?
Yeah, skin explants are not readily available, and there are many countries that have strong regulations with regard to the explants. I know in France, for instance, it's relatively easy to get this type of the material, but in some other countries, it's not as easy, or in some countries, even impossible to buy this type of the material. In Brazil, for instance, as far as I heard, it's not really possible to buy skin for some kind of research, commercial research, and so on. So, and the second thing is that these reconstructed tissue models are really much more standardized. They are representing the, let's say, young, average skin with relatively well-developed barrier. And the skin explants are every time different. It is really a hugely variable material. And even in the guideline 428, that is also used in the pharma field. The pharma people specified that they are not allowing the human skin for this type of the test. They're using pig skin because it is less variable. Human skin is highly, highly variable material. This is what I heard.
Okay. Are you okay, Nathan? Would you want to complete the question or no, satisfied? Okay. So another question from Casturi. Thanks for an excellent talk, Helena. what pointers would you have for young startups that has developed a skin model for cosmetics, pharma, industry, medical device, and are ready to submit data for regulatory consideration?
Okay, so if you have well-developed skin, you need to publish. You publish the barrier properties, histology. The easiest way how to show that it works well is to run the test for the skin corrosion with the reference material that are mentioned in the OECD test guideline, because this is already telling quite a lot about the barrier properties of the tissue and the readiness to be used for some more standardized uses. But you need to be quite clear what you want to do with your tissue model, because the competition on the market is high. So I tell you, you would not survive commercially if you just count on the skin corrosion testing. So you need to find a good use of your tissue model. And I would recommend you to make it a bit different for a different purpose. Yeah, not everything on the market, it doesn't need to be developed all for the regulatory testing. There could be models that have something more specific, like, I don't know, hair implemented, or vascularization, or immune cells implemented, or it can be model of the aged skin, sorry for my English, aged skin. And you need to find a niche in the market that is not yet well addressed by the other big guys that are doing these things for 20 years. You can also approach a pharma company, big pharmas, and offer them to develop something very specific for them. So there are many ways how you could place your novel 3D tissue model on the market.
Thank you. That was a very complete answer. So another question for Middle Miss. Hi, Helena. Thank you for your informative presentation. ISO 10993/23 currently states that for special irritation tests relevant for medical devices intended to be applied to a specific area, mucosal or eye or epithelia, the ARHI models are not adapted, and it is recommended to explore the use of other in vitro models with relevant cells or tissue if qualified for use with medical device. Do you know if this being investigated further to qualify the ARHI model for mucosal irritation with regards to medical device extract?
Yes, I have positive answer for you. This is further investigated. There are already publications of several groups targeting the eye irritation or ocular area. So yes, we will also from our own group publish very soon a paper in ATLA on the ocular irritation testing, and another paper is in preparation for the oral mucosa testing. So yes, there are groups that are working on this type of the novel protocols that could be implemented. And I also can tell you that, in my opinion, the guideline should be slightly changed with regard to the wording, because some people have doubts about testing neat products. Also, neat products can be tested on the 3D tissue models, not only extracts. We know that, and I think that currently, as it is worded, at one place it says it is possible, and in other place it is not recommended. So definitely when these new protocols will be out, and they will be out quite soon, I think that we will need to change this type of the wording.
Thank you. I have two more questions. One from higher date. As you have explained, initially aim for guidelines developing was too close to animal models. Should we expect now new human data-based guideline version of existing guideline?
I don't think so. We are happy that these things are implemented, and basically, even if they are closer to the rabbit data, you know, the results that you get with these old validated protocols are protective towards the human health. We know today that if we would keep the 15-minute protocol for epiderm as we have it for the episkin, we would have better prediction towards skin irritation that we see in human. But nobody really wants to reopen this anymore because, yeah, it's just would be another level of the complexity. But Fortunately, changes were in the minds of many people, and human data are today very well accepted for the, providing evidence that this really works, or this is a better model or better protocol than what we currently have in the animals. But, in the time when we are conducting these skin irritation and skin corrosion validation studies, there was no discussion about the human data. People didn't want to see it because, yeah, there were different reasons for that. But now, of course, we are 20 years later, and there was a progress in thinking. And now the human data are, of course, important part, and usually are also considered when people look into the overall sensitivity and specificity, especially in the cases that something really don't fit into the overall in vivo in vitro picture.
Thank you very much. So one question for Amata Misra is your printed full thickness skin will be more relevant for phototoxicity testing?
Depends whether 3D printed or done by the lab technician. It really doesn't matter so much, I think, for the phototoxicity. Phototoxicity is caused basically by radical reactions, free radicals in the skin. And if the skin can react properly, then of course you have possibility to test the phototoxicity. If the bioprinted full thickness model has something better or something more relevant, what the classically produced tissue model don't have, then it might be better, but it needs to be really checked and tested. As far as I know, these simple models are quite well predictive of the phototoxicity, and it has been shown in many studies that the phototoxicity predictions are really quite accurate. What would be needed, and there is, if someone would like to explore this, we would really love to enhance the OECD test guideline 498 by the potency testing. So right now, the phototoxicity guideline tells you whether material is irritant and non-irritant. But we know that we can also address the potency of the phototoxic material, but there are not enough studies conducted to convince some of the experts on the panel. So if you would like to help, please produce some data on the potency, regardless of model that you have, and publish it, because this will be extremely helpful to enhance the applicability domain of that guideline.
Thank you. So the last question, Sofia Poulou. Thank you, Helena, for the detailed presentation. What is the limit for acceptable sensitivity and specificity for validation? Is the standard for sensitivity and specificity based on animal data or human data?
Yeah, so it technically and historically, in the studies that I presented, it was based on the animal data sets, and for the sensitivity and specificity for the skin corrosion and irritation, the borders were set like something like 80% sensitivity and specificity for the skin corrosion and irritation. For the eye irritation, it was a slightly different story. There were big analyses done. It was sensitivity of 90% and specificity could have been lower because the eye irritation works with more than one method. So it's really a matter of, what is the strategy for the testing and how well can the animal data set predict itself. There were some studies conducted by Sebastian Hoffman and some other groups that showed that even the animal test cannot reproduce itself when the testing is repeated. So it could not reach the 100% conclusion on some of the materials. Yeah, so technically, historically, it's the animal data, and I hope that in the future, much more human data, if they are produced in the standardized way, could be accepted.
So thank you, Helena. So it is 1 p.m. So I think we have no more questions. There's been many questions. So thank you very much for your very excellent presentation and your very precise answer to all the questions. I think people in the chat, thank you very much for the presentation, very informative, etc. So thank you again for your contribution to the EUROTOX webinar. And I wish you, Helena, a very nice afternoon, and wish you all a very nice afternoon. Thank you again for attending. Thank you.
Thank you. Bye-bye.
Bye-bye.